and covers the Quadrat practical work required for Unit 4, Ecology and the Environment. This is a quadrat. They are traditionally square but can be different shapes. They are used to mark out an area to investigate. They can be commercially made, or in this case, due to being in lockdown, they can be made from other materials such as wire coat hangers. These quadrats are placed on the ground to look at plants or slow-moving animals within them. When using quadrats, we can look at various things. The total number of individuals of one species, for example dandelions in an area. This is the first type of sampling we're going to do today. Or we can look at the species richness, which is a number of different plant or animal species in an area, but we are not looking at the number of individuals within a species in an area. Or we can sample the percentage cover, the percentage of the quadrat area that is covered by one species, for example grass. This is easier to estimate if a quadrat has wires marking smaller sections. Some quadrats are broken down into 100 individual squares, which make this much easier. Percentage cover rather than number of individuals is used when estimating plant frequencies if it is difficult to identify individual plants.
Percentage cover is the percentage of the quadrat covered by one species. It tends to be a faster way of collecting data, but it is more subjective. You simply estimate what percentage of the area enclosed by the quadrat is occupied by each species. In this example above, what we have is a quadrat which has been broken down into 100 separate squares, so it's 10 by 10. This means that each square represents 1%. Here the moss covers 7 whole squares, marked 1 to 7. And then there are 5 squares where it covers more than half of each of those squares, so they are also counted as percentage. The unmarked boxes here, which still have green in them, are not actually counted because they are not covering more than half of the square. Therefore, 7 whole squares plus 5 almost whole squares equals 12%. So we would record moss, percentage cover, 12%.
We can use these ways of sampling to compare two different areas which have different conditions or are in different countries, and they are often used to investigate why an organism prefers one area to another. Quadrats can also be used to measure biodiversity of areas. As I have mentioned in my previous lesson, biodiversity is used to describe the range and variety of living organisms in an ecosystem. Biodiversity looks at the richness, the number of different species in an area, and the number of each species and whether the distribution of each species is spread evenly in the ecosystem. As explained before, biodiversity is important for many reasons, and human activities are having a huge impact on it, so studying areas and recording changes in biodiversity is very important to try and protect the environment. For the Dual Award Science, you just need to know about investigating the population size of an organism in two different areas. Today we are going to look at the distribution of dandelions in a wood and in an open area. If you were asked whether there were more dandelions in the wood or in the open area, you could either try to count every plant in each area, which would take a very long time, or you could take detailed samples from set areas using a quadrat like this. Take the mean of each quadrat, then multiply the number up. I'm using a 1/4 metre squared quadrat. I will take 10 samples in each area, then add up the total number of dandelion plants found in those 10 quadrats and divide by 10 to get the mean number of dandelions in a 1/4 metre squared. Then, because I've marked off an area which is 100 metres square, I will multiply the mean by 400 to give a good estimate of the total number of dandelions in the area. The area will be marked out using these tape measures, which will be pinned down at right angles to each other. They act like an X and Y axis. Each quadrat should be placed randomly so that a representative sample is taken. So I'll use random number tables that I have here to determine the coordinates of where each quadrat will be sampled from. The first random numbers are 7 and 3, so I'll ask my filming assistant to stand at 7 metres on the y-axis and I will find 3 on the x-axis, and I will walk out until I'm in line with her. I will then put the bottom left-hand corner of my quadrat at that coordinate and carefully count the number of dandelions in that quadrat and record it in this table. I will also record the light level, using this, at each quadrat to compare with the wooded area. I will then repeat this 9 more times so that I get 10 results. It is important to get repeats like in all experiments. It makes your results more valid and reproducible.
I now need to show you a dandelion plant, as I know some people may not know what the leaves look like, even though most of you will be familiar with the bright yellow flower. Dandelion leaves generally have toothed edges like lion's teeth, which gave the plant its name from the French dandelion. The leaves are hairless, and along with the hollow flower stem, as you can see, hollow, there we go, and they all grow directly from the rootstock, which is right down here at the bottom.
Seven meters, ready.
Okay, I'll find 3 meters then. There's 3 meters. So I'm at 3, you're at 7, so that's the coordinates. So now what I've got to do is walk out as straight as I can until I'm in line with you. Right. I'm going to put my quadrat down there as I explained, and then I need to take, first of all I will turn my light meter on, and I have got, let's put it down at the quadrat. That is, without anybody getting in the way, 87,500. 87,500, that's that done. So now I have to look at the number of dandelions. So I've got 1 there, I've got 2 there, I've got 3 there, little one. Don't miss the little ones because their leaves aren't as jagged, so they're not quite as easy to see. So 1, 2, 3, I've got another one, small one there, 4, another small one there, 5, and 6. So I've got 6 dandelions altogether. So I record that in my table, 6 dandelions, light intensity is 87,500, and then I'm going to do my other 10. No, my other 9. This is my first one, I need another 9, and then I can take a mean.
When counting plants, you only count plants that are more than halfway into the quadrat, as in the diagram above. The next few slides show photos of the other quadrats, quadrats 2 to 10. You need to draw up a results table for the open area like the one above. The next 9 photos then show the other quadrats from the open area. Using the data in these photos, you then need to complete your results table. Quadrat 2 has already been done for you. Your results table should look like this. You've completed recording data for the open area, so now we are going to look at the wooded area.
This is the wooded area. I'll set up the 10 metre by 10 metre area as before, using the tape measures at right angles. I will then use my random number tables to get the coordinates for where I will place each of my 10 quadrats, and I'll record the number of dandelions in each quadrat. And of course I've got to check the light intensity at each quadrat and record that in my table for analysis later. I'll take photographs of each quadrat so you can then record your own data for further analysis.
Now you need to try completing a results table for the wooded area by looking at each of the quadrats yourself and seeing whether you can count the number of dandelions. It is much easier with these quadrats as the dandelions tend not to be hidden by grass. I suggest you pause on each of the quadrats before watching the video that follows to check your answer. Make sure you record the light intensity from each of the photos too.
Quadrat 1 then. Hopefully you all managed to see it was just one big dandelion. Quadrat 2 then. Again, there's just one big dandelion. This one had a leaf in there, but it doesn't count, so it's just one. So, as you can see, there's absolutely none in there, so 0. Quadrat 4 then, absolutely none again, so 0. So
This is Quadrat 5, and again we just have one dandelion. So again, no dandelions. So there's one dandelion in this. Quadrat 8 then, we have this one here. This one doesn't count because it's just the leaves. But we've got this one, and just over half its rootstock is in the quadrat. So we're going to count that as a second one. Quadrat 9 then, we've definitely got one there, 2 there. Oh, and we've got another one there, so there's 3. Last one, couldn't be easier, could it? There is no dandelion in there.
Your results table for the wooded area should now look like this. Now you need to add up the total number of dandelions for all 10 quadrats and divide by 10 to get a mean. Then add up all the light intensities for all 10 quadrats and divide by 10 to get a mean. Enter them in the last column. Now you need to do the same for the open area. When you add up all the number of dandelions in the wooded area, you should have ended up with 7. So you divide that by 10 and the mean is 0.7. When you add up all the light intensities, you end up with 7,035. If you divide that by 10, the mean comes to 703.5. When you look at the open area, the total number of dandelions is 57. So the mean is 57 divided by 10, which gives you 5.7. And with your light intensity, you should have ended up with a mean of 85,550. Make sure all means are to the same number of decimal places. As you can see, there are definitely differences here. Why do you think there are so many more dandelions in the open area? Can you come up with a conclusion to this? Have a think and we will come back to this. We still have to estimate the number of dandelions in each area. So the mean number of dandelions we have calculated so far are for one quadrat measuring 0.5 metres by 0.5 metres, which is 0.25 metres squared. But the area we were looking at was 10 metres by 10 metres, which is 100 metres squared. If you divide 100 metres by 0.25, you get 400. This means that 400 quadrats could fit into this area. So to find the estimated number of dandelions for the whole area, you need to multiply the mean by 400. For the open area, this means it is 5.7 times 400, which equals 2,280 dandelions. And for the wooded area, it is 0.7 times 400, which is 280 dandelions. This data could then be represented in a bar chart with, on the x-axis, the different areas, the open area and the wooded area, and up the y-axis, the number of dandelions. You could plot the results collected from each of your 20 quadrats on a scatter graph, putting the light intensity on the x-axis and the number of dandelions on the y-axis. This gives you an indication if there is a correlation between the light intensity and number of dandelions. Remember to use the rules for drawing a graph. I use St. Paul, where S is for scale, making sure the graph covers as much of the graph paper as possible, at least 3/4 of the page, and that the numbers go out linearly. A title to explain what the graph shows, for example, graph to show the number of dandelions against light intensity. Plots, make sure you use a sharp pencil and nice neat crosses. The axes, make sure they are labelled. Units, all units must be included, so here lux for light intensity. Lines, use a sharp pencil, one continuous line, not sketchy, and a line of best fit so you are leaving approximately the same number of plots above the line as are below the line. Plants need light, water, space and mineral ions to grow well. Light energy is needed for photosynthesis, and water is a raw material for photosynthesis and is also needed for support of the plant, transport of substances around the plant, and for metabolic reactions. If the light energy is being blocked by trees, and we saw this in our results when measuring the light intensity, then less photosynthesis can occur, and less glucose is produced in photosynthesis, so there is less for respiration and less for making other molecules such as protein for growth. The trees also take up a lot of the space, and their root systems are extensive, taking up water and minerals from the ground. We did note that the dandelions in the wood area grew taller because they are trying to reach areas where light breaks through.
For the separate award science, you also need to investigate the distribution of organisms in their habitats and measure biodiversity using quadrats. To measure species richness, we will count the number of different species in each quadrat in the wood and in the open area. This gives an indication of biodiversity. If you go on to do A-level biology, we will use species richness and species evenness, the number of each species in each area, to calculate the Simpson's Index of Diversity, which is a statistical test to compare biodiversity. To measure the richness, I will carry out the placing of the quadrats using random number tables as before, but this time not just looking at the dandelions, but looking at the number of different species. It is great if each species can be identified, but they don't need to be, and can just be called species A, species B, and so on, as we are only interested in the number of species. We will take the top 10 quadrats from each area and record the number of species in each quadrat so we can compare species richness between the areas.
To investigate biodiversity in the open area, you will need to draw up a results table like this. Then watch each video and place a tick next to the plant when I spot it in a quadrat. Remember, we are only recording if the species is there, not how many of each are present. I have written down the main plants in the quadrats, leaving other for any extras. Then you can check your results for each quadrat and record the light intensity on the photo that follows each video clip.
Number 1. So we've got dandelions. We've got this grass. We've got a different type of grass here, so that's another one. What else have we got in here? Have we got anything else? No, is that, yes, that's a different type again, so there we go, we'll give that one. But that's the same as that one. So there we go, we've got 4 different species in there. Quadrat 2 then, there's this one, so that's one of them. There's obviously dandelion here. There's the normal grass. No, that's not actually in the quadrat, even though the leaves are, it isn't, but this one is, and that's the same as that one. So that, is there anything else I can see? What's this one here? Oh yes, that one's different, I think. Yes, there's clover down there. That's 5. Okay, 5. This is Quadrat 3. We've got this type of grass that you can see, the prominent one here, which has got a soft hairy leaf. And we've got the dandelion. What else? Oh, we've got the vetch, but does that grow from, no, that grows from outside the quadrat, so we can't count that one. Only things that are growing or rooted in the actual quadrat are we going to use. Bare earth there, obviously. There's that obvious grass, and then there's this other fine grass, so that's 3 different species here. Quadrat 4 then, there's this plant here. Ooh, it's got cuckoo spit on it. And there's a dandelion. And there's clover, there's white clover there. So then we've got this soft grass again. Then we've got the fine grass at the bottom. There's more clover there. What else have we got? Got anything else? And that's it, I think. Quadrat 5 then. We have this soft grass again. Got dandelions. What else can I see from up here? We've got the normal grass, the foundation grass at the bottom there. Now, did I say clover? I didn't say clover, did I? No. Okay, that's it then.
Quadrat 6 then, you can obviously see the clover there and the dandelion here, so that's, we're used to those, and the normal foundation grass down there, and different type of grass there. Have we got anything else? That's it. So Quadrat 7 then, slightly easier to see, but there's quite a variety here. There's one there, 2 with the clover, 3, 4 for the foundation grass, 4, 5 with the dandelion, different grass again, a soft one, so that's 6. Is there anything else? It's a question, because there might be. Because there's a little bit more sun. Okay. Quadrat 8. Now the flies have stopped pestering. Being the best of my film crew. Right, 1. That vetch, that climbing one there. 2. Dandelion 3. There's quite a bit of that vetch there. There's this one, which is 4. Is there any clover in there? No clover. No clover on this one. And then there's the common grass at the bottom, which is 5. Alright. Quadrat 9 then, we've got this, I think it's a vetch climbing one there, that's 1. And you've got this one, which is 2. Got the ground grass, which is 3. We've got the fluffy grass, which is 4. Dandelion, which is 5. That's all part of the same type of grass. Okay. Quadrat 10. We've got the ground grass and the dandelions, which is what we expect to see. We've got the soft grass. We've got the dandelions. You should have a results table that looks something like this. You can now calculate the mean number of species found in a quadrat in the open area and the mean light intensity in lux. We will now look at the wooded area. It is slightly easier to spot the different species in each of these quadrats, so this time I will show you the photos first. I suggest you freeze each one to get a careful look at the plants. Record what you think you can see and record the light intensity, then check your results with each following video. Quadrat 1 then. It's the grass and moss. Oh, and this little chap here. So, 3. Quadrat 2. We got him that I'm not touching. This little one here, 2. Which is the same as that one. The moss, which is 3, and this one that's in here too. That's 4. Quadrat 3, that one. Oh, a dandelion there. And 2 and 3. And that's different, so that's 4. Have we got any moss? No moss. So 1, 2, 3, 4. So this is also, ooh, it is sticky weed. Quite a bit of that. So that's 1. Then the grass, which is 2. The moss, which is in there, which is 3. Not going to touch that, it's horrible stuff.
Let's see if there's anything hidden under it. Oh yeah, there's a little plant trying to come up there. That's 4. Four. Okay, 4. Quadrat 5. Big dandelion. That's 1, and a little plant here. 2. Try not to get prickled. Sticky weed. 3. That's the same as that. That's the same as that. Moss. 4. No grass. Okay, 4. Quadrat 6. Lots of grass and moss and dandelion. A little one there. Okay. Quadrat 7 then. There's this. That's 1. Dandelion 2. Moss 3. Ooh, that's not dandelion, like I said. That's it, 3. Right, this is quadrat 8. So we've got a couple of big dandelions there and some sticky weed. It's moist down there. But that's it. Is it? No, it's a moss. Yes, there's a bit of moss there. So there's another one there which is moss. Anything else? Oh, okay, 3. This is quadrat 9 then. It's a lot lighter here, much lighter. This is a dandelion and sticky weed and moss. Dandelion, sticky weed, moss. What else have we got in here? Have we got anything else? Apart from sticky weed, moss and dandelions. And that sticky weed really is rather sticky because it won't stop sticking to me. There we go. Quadrat 10. So we got one there. We've got the dandelion there. We've got the prickles. Oh no, I don't want to get prickled. Moss down there, which is 4. Ooh, I don't want to touch the prickle. That's dandelion. Ooh. Getting poked in the face. Right. Ooh, you get poked in the face. Right. Did I say moss? Yeah.
Moss 1, 2, 3, and moss, which is 4. 1, 2, 3, 4. Good. 4.
Hopefully you ended up with a results table that looks something like this. Again, try calculating the mean number of species and the mean light intensity for the wooded area. In the open area, the total number of species is 45, so to find the mean, divide by 10 as there were 10 quadrats, and this equals 4.5. The mean light intensity is 86,600 lux. In the wooded area, the mean number of species is 3.6, and the mean light intensity is 4,571 lux. This indicates that the open area has greater plant biodiversity, which is likely to be due to the fact there is more light availability for photosynthesis, as there are no trees blocking the sunlight to the ground. The difference is not large, but there is a difference. Now try plotting your results on a bar graph, where you put the area at the bottom on the x-axis and the mean number of species up on the y-axis. We have now finished the work from Unit 4 on quadrats. During the lesson, we have described the use of quadrats for sampling the distribution of organisms. We have estimated the population sizes in an open area and a wooded area. We have measured the biodiversity in an open and wooded area, and we have discussed the use of bar graphs and scatter graphs as a way of presenting data, making sure we follow the rules of graph drawing.